Hello everyone, this is the last video about general and special senses. And now we are going to cover equilibrium and hearing. We can anatomically divide our ear into three distinct regions. We have the external ear, the middle ear, and the inner ear. The external ear contains the auricle, the external acoustic meatus, which is the outer ear canal, and the external side of the tympanic membrane. Now, the middle ear is this region here in the middle, and it's sort of an air-filled cavity, which has the inner side of the tympanic membrane, and this side of the tympanic membrane is associated with the auditory ossicles, which are a series of bones called malleus, incus, and stapes that transmit vibration for sound. Also in the middle ear, we find the opening to the auditory tube. This tube is basically an elastic tube that connects the middle ear to the upper part of our throat, which is technically named nasopharynx. After the middle ear, we have the inner ear, and the inner ear is really a bony labyrinth located within the temporal bone. In the inner ear, we find the sensory structures associated with equilibrium and hearing. And these structures are the vestibule apparatus, which is the structure involved with balance and equilibrium, and the cochlea, which is the structure involved with sound. Now, what we see here leaving the vestibular apparatus and the cochlea is the vestibulocochlear nerve, which is cranial nerve number 8. Starting back with the external ear, we have the auricle, which is sort of the elastic cartilage of our ear that leads to the external acoustic meatus, which is the outer ear canal, and that ends at the tympanic membrane. Within the external acoustic meatus, we find ceruminous glands that secrete cerumen, which is basically earwax. These ceruminous glands are modified sebaceous glands, which create earwax, and because it's wax, it's hydrophobic, and it can help to keep water from staying inside of our external acoustic meatus, and thus preventing infections. The middle ear is like an air cavity just deep to the tympanic membrane, and it is the place where we find the auditory ossicles, which are the three smallest bones of our entire body, the malleus, incus, and stapes. So MIS, malleus, incus, and stapes. The purpose of these three auditory ossicles is to amplify the sound waves, so that when the sound wave travels through the external acoustic meatus and reaches the tympanic membrane vibrates, and this leads to the vibration of the malleus, incus, and stapes, which amplify the sound wave. Now the stapes, the last one of these three little bones, vibrate against the oval window of the cochlear canal, and the cochlea transduces these vibrations into sound. So the purpose of these three ossicles is to amplify sound waves, and that's the reason we can hear when someone speaks very low. Now, this amplification can become a problem if we are exposed to very loud sounds, noises. And in this case, we don't want to have the sound waves amplified. And that's the reason why we have these two skeletal muscles, the tensor tympani and the stapedius, in our middle ear. These muscles control the ability of the ossicles to vibrate. So when these muscles are contracted, it's harder for these ossicles to vibrate, and the sound waves do not get amplified as much. And if these muscles are relaxed, it's easier for the ossicles to vibrate, and the sound waves get amplified as much as possible. As you can see here, the middle ear is connected to the auditory tube, which opens up to the nasopharynx, which is the upper part of our throat. This tube helps equalize the pressure that's inside of the tympanic membrane to the pressure on the outside of the tympanic membrane. Now, an obvious question is why wouldn't the air pressure on the outer ear be the same as the middle ear? And a good example is when the airplane takes off and the air pressure changes inside the cabin of the plane. With that, the air pressure on the outside of our body becomes different than the air pressure in our middle ear. And when that happens, we experience an uncomfortable popping sensation, or we feel like we are talking on a barrel. And then we see some people chewing gum, opening their mouths very widely, and some of them pinch their nose and inflate their cheeks. All of that is to force air from the throat through the auditory tube in the middle ear, so that the air pressure in the middle ear returns to being the same as on the outside of the body. And the moment the air pressure in the middle ear becomes the same as the pressure on the outside of the body, that popping sensation goes away. Now, there is a downside of this tube connecting the throat to the middle ear. And the problem is that this tube is related to the middle ear infections, which is what we know as earache. So what happens is that if we have a sore throat, this infection can spread up the auditory tube and into the middle ear. And when that happens, we have an infection of the middle ear, and that's clinically known as otitis media. Okay, so now going deeper, we have the inner ear. And the inner ear exists within a bony labyrinth, within the temporal bone. And what I mean by a bony labyrinth is basically convoluted spaces within the temporal bone that houses the membranous labyrinth. And the membranous labyrinth is actually the structures involved with equilibrium and hearing. Here we have a detailed view of the membranous labyrinth. So if we zoom in a cross-section of the temporal bone, we can see these odd-looking structures deep within the temporal bone. And the spaces that house these structures is called bony labyrinth. So within the bony labyrinth, we have the membranous labyrinth, which is all this shown here. And the membranous labyrinth includes several structures, such as the cochlea, which is this one that looks like a snail shell, as well as the semicircular ducts and the vestibule. It turns out that between the bony labyrinth and the membranous labyrinth, there is a thin fluid-filled space, and this space is filled with perilymph. Now, if you recall, peri means outside, periphery, and lymph is like a fluid, an aqueous solution. So perilymph is what helps preventing the membranous labyrinth from rubbing against the bony labyrinth. Now, within the membranous labyrinth, we have the endolymph, and endo means within. And this is the fluid that we find filling the membranous labyrinth and helping to support it from the inside. Now, we can divide the membranous labyrinth into three main regions: the vestibule, the semicircular ducts, and the cochlea. Within the vestibule, we find a pair of membranous sacs called utricle and saculo. And projecting off the vestibule, we find the three semicircular ducts. The vestibule and the semicircular ducts together, they form what we call vestibular complex, and they are involved with balance and equilibrium. Now, the cochlea that you see here, that looks like a snail shell, is not involved with equilibrium. It's actually involved with hearing, because it contains specialized cells that transduce sound waves into action potentials, and that allows us to become conscious of sound. So inside the cochlea, we have the so-called cochlea duct. And inside the cochlea duct, we have endolymph that I just mentioned. Zooming in into the cochlea to have a more detailed view of the cochlear duct, we can see that the cochlear duct lies in between two chambers, the scala vestibule and the scala tympani. these two outside chambers are filled with perilymph, and they are just there to support our cochlear duct. Now, the cochlear duct is filled with endolymph, and inside the cochlear duct, we find the spiral organ. And one important detail is that this spiral organ extends through the length of the cochlear duct, going all around until the end of the cochlear duct right here in the middle. Now, if we zoom in the spiral organ per se, we can see that we have hair cells, which are the auditory receptors, also called cochlear receptors. The hair portion of these cells extends up and touches a membrane called tectorial membrane. So what happens is that when sound gets transmitted to the cochlea, it causes a wave in the endolymph inside the cochlear duct. And this wave leads to the vibration of the hair cells against the tectorial membrane. The vibration of these hair cells causes the hair cells to generate action potentials, which gets transmitted down to these axons and continue to go to the vestibular cochlear nerve, and that sends the information to specific regions of our brain, which interprets as a specific sound. Now, I just said that the wave of endolymph inside the cochlear duct is what leads to activation of the auditory receptors. I would like you to imagine a pool, a plastic pool filled with water. If you start tapping on the pool wall, you will cause the water to start moving like waves. Now, the shape of the waves that you create is going to depend upon the frequency that you are tapping on the pool wall. If you tap slowly, you create kind of long waves. And if you tap fast, you create short waves. So what we can see is that different frequencies can create different wave patterns on the water that we have in the pool. So the point that I'm trying to make is that different frequencies will also cause different types of waves on the endolymph inside of the cochlea. What happens is that the same way as the waves you created by tapping on the side of the pool, a high-frequency sound creates short waves in the endolymph, and this short wave causes the bending of the little hairs near the beginning of the cochlea. And then these are the cells generating action potentials that get transmitted to the sensory neurons that send this information to our brain. And when our brain receives these signals, our brain interprets that as a high-pitched sound. Now, the low-pitched sound is like it has a lower frequency, and that creates longer waves. And this long wave in the endolymph fluid bends the hairs of these hair cells located more deep inside the cochlea. And when this signal is sent to our brain interprets as a low-pitched sound. And this is what you're seeing here. High-frequency sounds cause short fluid waves, and these are detected by hair cells located closer to the beginning of the cochlea. Now, on the other hand, low-frequency sounds creates like long waves, and these long waves are detected by hair cells located closer to the end of the cochlea. That's right here in the middle. So let's do an overall summary of all what I just said. We have sound waves, which are generally they're basically waves of air pressure, right? And they travel through our external acoustic meatus. And then the sound wave will bounce off the tympanic membrane and cause the tympanic membrane to vibrate. Then the vibrating tympanic membrane will cause the vibration of the malleus, that vibrates the incus, that vibrates the stapes. And the stapes vibrates the oval window, which is a membrane at the beginning of the cochlear canal, the cochlear duct. Now, this vibration right against the oval window creates waves in the endolymph inside the cochlea. And that vibrating endolymph travels along the cochlea until it reaches a specific patch of the tectorial membrane. And there we have the hairs getting bent. When the hairs bend, it generates action potentials. Now, these auditory information in the form of action potentials travel through the auditory branch of the vestibular cochlear nerve back towards to the brainstem, and it connects with the mesencephalon, which is involved in auditory reflex and sound localization. And if you recall, when we learned about the mesencephalon, I told you that the mesencephalon was related to the unexpected reflexes in response to sound. So, like, we are in the mesencephalon, and then from the mesencephalon, the action potentials go via axons to the thalamus, and the thalamus will send the information to the temporal lobe, where the primary cortex, auditory cortex, is located. Going back to our cross-section of the temporal lobe, we see that besides the cochlea, which is a structure responsible for detecting sound, we have these other structures called vestibule and semicircular ducts. And together, the vestibule and the semicircular ducts form the so-called vestibular complex, which is involved with balance and equilibrium. Here we have a diagram showing more clear the vestibular complex. Within the vestibule of the vestibular complex, we find two membranous sacs called utricle and saccule. The utricle is oriented horizontally, and the saccule is oriented vertically. You don't need to know which one is which. The purpose of these two sacs is to inform us about gravitational pull and linear motion. So, in other words, it tells us when we are speeding up, accelerating, or when we are slowing down, decelerating, in a linear direction, not in a rotational direction. Now, on the other hand, the semicircular ducts, which are these semicircular structures projecting off the vestibule, are involved with informing us about rotational movement in space. So, now let's zoom in into the utricle and saccule, which are the membranous sacs I just mentioned. So these sacs, you can see here, one of them, in the case is the utricle, and we have something called otoliths. And otolith is like otto means ear, and light means rock or stone. So otoliths are little rocks. And these little rocks are within a gelatinous layer, which form what we call the otoliths membrane. Now, guess what we find inside the utricle and the saccule? We find beautiful hair cells. And these projections of these hair cells are in contact with the otolithic membrane. So, as you can imagine, when we move in a horizontal or vertical direction, gravity causes the otolithic membrane to move, which causes the hairs to bend, and when these hairs bend, they generate action potentials, which get transmitted along the vestibular branches of the vestibular cochlear nerve. So what it looks like in action is that when we are standing upright, the otolithic membrane inside of one of those little sacs, the one that is oriented horizontally, which is the utricle, this otolithic membrane is not bent, and it's sort of horizontal due to forces of gravity. Now, also due to gravity, once we tilt our head, gravity causes the otolithic membrane to move in the direction of gravity and cause the hairs in the hair cells that are embedded within the otolithic membrane to bend. And when these hairs bend, it generates action potentials that are transmitted to our brain via the vestibular branch of the vestibulocochlear nerve. And then we feel like we are tilting our head forward or sideways. Now let's imagine we are in an elevator moving up. In the sac that's oriented horizontally, the otolithic membrane is unbent, and it's sort of horizontal due to force of gravity. Now, the other sac that's oriented vertically, which is the saccule, because it's oriented vertically, when we start moving up, it will cause the otolithic membrane to move downwards. And when that happens, that causes the little hairs to bend, and that generates action potentials that are transmitted to our brain. And we feel that we are going up in the elevator. Now, what's interesting is that the utricle and saccule allow us to feel acceleration and deceleration, but they do not allow us to feel constant speed. And that's the reason why when the airplane takes off, even if we have our eyes closed, we feel it. And when the airplane is landing, we also feel it. But while you are flying, like at 600 miles per hour, we do not feel it. We do not feel constant speed because the otolithic membrane inside the utricle and saccule is not moving. So you can just know that you're moving because you see the clouds passing by. Okay, so we just learned about the utricle and saccule, and they are the ones that inform us about linear motion. But we can also feel rotational motion. And the structures that allow us to feel, to be aware of rotational movement are the so-called semicircular ducts. The semicircular ducts are called semicircular because they are actually semicircles that project off the vestibule. And we have the posterior, anterior, and lateral semicircular ducts. And we should think of them as X, Y, and Z, which gives us the three-dimensional representation of our head in space. Now, guess what we find inside the semicircular ducts? We find hair cells, again. So look at this. We have these hair cells within the semicircular ducts that detect rotational movement. Like when we spin to the right or we spin to the left, for example. Even if we have our eyes closed, we are aware, we are able to feel that we are spinning. And that's because when we are spinning, the endolymph inside the semicircular ducts move in a direction, in a specific direction that we are moving. And that causes the band of the hair on the hair cells within what we call ampullary capule. And you don't need to know the name of this ampullary capule. But what you need to know is that the bending of these hairs leads to the generation of action potentials that are transmitted to our brain via the cranial nerve number 8, the vestibulocochlear nerve. So, since we have the semicircular ducts in the X, Y, and Z directions, it gives us the three-dimensional representation of our head. And we have the sensation of moving in all directions, even when we have our eyes closed. In other words, that's why when we are in a roller coaster and we get scared and we close our eyes, it doesn't help. Because even with our eyes closed, we feel everything. Now, another interesting thing is that if I'm spinning you, and then I stop very quickly, you will still feel like you're spinning. And the reason for that is because the endolymph inside the semicircular ducts, it continues moving. And we just feel like we stop moving when the endolymph stops flowing. Because while the endolymph keeps flowing, the endolymph keeps bending the hair cells, and that leads to generation of action potentials that inform our brain that we are still spinning. So we just stop feeling that we are moving when the endolymph inside our semicircular ducts stop moving. This diagram just shows us the vestibular branch of the vestibulocochlear nerve that takes the sensory information from the vestibular complex, the utricle, saccule, the semicircular ducts. It takes the information back to the brain, to areas like the cerebellum, thalamus, and cerebral cortex. And when the information, the action potentials, gets to those areas of our brain, it allows us to become aware of our head position in space. So with this, we finish the general senses and special senses chapter. Please let me know if you have any questions. I will see you in class. Bye.